Okay, guys, so this lecture is going to cover the immune system. There's two parts of the immune system, the innate immune system and the specific or the adaptive immune system. So we're going to talk about both parts of them, kind of what they are, kind of how they work, some of the things that are associated with the components of the immune system, some of the responses the immune system has to different pathogens and things like that. So let's go ahead and get started with a little bit of history of how the immune system was discovered. So this guy right up here, Ilya Michkinov, a Russian guy, he got to looking underneath things underneath his microscope, kind of one of his passions. And he kind of had this idea that there was something in the body. He wasn't sure what it is. It was some sort of cell. He wasn't sure what type of cell or anything like that, what it looked like. But he just predicted that there was some sort of cell in the body that served to eat bad things. So they knew at this point in time that bacteria were able to cause disease. Um, so he predicted that there was something inside of the body, some sort of cell that would eat these bad bacteria, these bad things that got in the body, that protected the body from infection, so the bacteria didn't cause problems. He didn't know these things existed. He couldn't prove that they existed at the time. So he just published a paper about his idea. This is the hallmark of a really good theory. You can write an idea about what it is, make a prediction, and then years later, he confirmed his ideas by looking at a tiny little water flea. Now you can see these guys down here, they're clear. So what he did was he watched this cell inside of this water flea here eat a yeast cell. So he termed these cells phagocytes, which means cells, sites who eat, phagocytosis, cells who eat. Um, the cells, the part of the immune system that destroy things. But he actually watched one of these cells eat a yeast cell inside of one of these little clear see-through water fleas. So he confirmed that these types of cells existed. So this was the very first step to identifying the parts of the immune system, the very first idea of the immune system, that there were things inside of the body, cells, that would eat other things inside of the body to try to protect us from getting sick. So, the study of the immune system is called immunology. Pretty easy, the immune system and the study of. The immune system does tons and tons and tons of useful things for humans. It's going to protect against the obvious, which is infectious agents and things like that. It can also protect against certain types of cancers, and also things like splinters, metal shavings and things like that get inside of your body. Different things like that. Your body can help protect against those. The immune system is also responsible for keeping your body safe if you have transplanted organs and things like that. So if you have someone who has a very similar immune system to yourself, I'll kind of touch on that a little bit. It's a little complicated. It doesn't really have a whole lot to do with microbiology. Um, but if you get into this concept of kind of the different markers on the organs and things like that that the immune system uses to read itself to make sure that it's the MHC complex, the major histone compatibility and whatnot, um, the immune system is going to read those proteins and things that are found on the surface of cells to tell if the organs that have been transplanted are a good or similar match or not similar match. So if you're very similar, your immune system doesn't really recognize it being a problem. And the less similar you are, the more likely the cells are to reject. The immune system is to reject that organ. Sometimes the immune system can freak out, have a problem, and it targets the cells of the person's own body or the organism's own body. And it's called an autoimmune disease. So self-immune, so autoimmune, the immune disease, the immune system attacks itself. It recognizes itself. That's not supposed to happen. The immune system is generally supposed to turn a blind eye to the cells that it recognizes as your own using that MHC complex idea. It recognizes your self cells kind of thing. And it generally turns a blind eye to cells that aren't recognized as your own. So sometimes the immune system, we'll talk about how this works in just a little while, can cause allergies and things like that. And different types of cells are associated with causing allergies inside of your body. So the immune system can be used in laboratory settings for lots of different purposes. You can blood type. That's how they tell if you're A, B, or O. Technically, that is A, B, or 0, the lack of a sugar, but it's just associated with O because it's an alphabet, so it kind of just makes more sense that way. But anyway, that's how they tell apart your blood sugars. But the sugar on your blood cells, the immune system reaction that they have is going to associate what type of blood you can have as a transplant. They're going to read the antibodies inside of your blood system, your immune stream, your immune system, your bloodstream. Sorry, switch those two words up. They're going to read the antibodies that are found floating around inside of there to help diagnose what diseases you may or may not have had, or what disease you have now, and things like that. Count your antibodies to see if you need a new vaccination or booster shot. And they're going to use how the bacteria actually interacts with the immune system, and things like that. To classify different species of bacteria. Different species of bacteria do different things to the immune system, or sometimes they don't even aren't even detected by the immune system. So that can be a species hallmark, how they interact with the immune system. And then you can kind of look at all this stuff together. What a person's blood type is, the different antibodies that they have in their bloodstream, the different types of antibodies, different levels of those antibodies. That can also help to identify unique blood markers and criminal markers in criminal cases and things like that. So you can kind of tell who's had what diseases and things like that. And it's a little more unique to people than just kind of like blood type or things like that. You can say, this person's got blood type and they've had certain such so and so diseases, and that's a little more helpful. The last thing, without a functioning immune system, even a slightly less functioning immune system, you can have a vitamin deficiency, or you don't have a healthy diet, or things like that, or maybe your immune system lacks a specific protein, it's got a problem, an autoimmune disease, anything like that. If your immune system can be off a little bit, even just the slightest amount, tiny little bacteria that a person with a normal, healthy, functioning immune system encounters on a daily basis, like Pseudomonas, Staphylococcus, Streptococcus, those commensalistic organisms, those normal flora organisms that don't normally cause problems in a healthy person with a normally functioning immune system. Someone without a healthy, functioning immune system, bacteria like that, they can cause some serious problems. The immune system's compromised. It doesn't work quite as well as it should. And even the simplest of bacteria can overwhelm a non-functioning immune system. So like I mentioned earlier, there are two different types of the immune system, or two different classifications, I should say. The innate, it's also called the non-specific, and the adaptive, which is also called the specific. So we're going to start off with the innate. We'll talk about the cells involved with it and how it works and things like that, and then we'll move on to the adaptive. So the non-specific immune system doesn't really have the ability to differentiate between bacteria species, viral species, fungal species, a splinter for that matter, tattoo ink, anything at all that's not supposed to be inside of your body. If it recognizes it as not your own cells, it will attack it, and it attacks it the same way every single time. It does the exact same reaction the same way every single time. Obviously, the reactions that it uses are going to work on some species. Since it does not change its approach at all, it's not going to work on other species. It's obviously not going to work at all on a non-living organism like a splinter, but it's going to try anyway. It just has the exact same reaction every single time. This is kind of like the ground troops, the first line, just send them in to get stuff done for the military. They don't really have any real specific training for kind of black ops or spec ops missions or things like that. They just go in, hit hard, and hit fast. And they hit everything they see with the exact same response. The specific immune system, on the other hand, kind of it acts a little bit more like a spec ops or a black ops kind of thing. It's part of your immune system. They have a tailored response to what they see. If they come in contact with a specific bacteria that part of the immune system recognizes, they will only attack that one specific part. They'll ignore the rest of them, and they will tailor their responses to what they see. If you see this bacteria and that one doesn't cause a concern to you, you're going to ignore it. But if this one does, you'll attack that one. But the difference is, this one, every single time you come in contact with a pathogen, the first time, the last time, every single time you come in contact with it, this part of the immune system is going to work. The non-specific, the innate immune system, innate meaning things that you're born with, which we'll get into in just a second, is going to have the exact same response every single time. The specific immune system, on the other hand, you have to come in contact with an organism or a pathogen the very first time for this thing to work. So the second time you come in contact with it, it won't work. The first time, or it will work, excuse me. The second time it will, sorry, let me start over for just a second. The first time you come in contact with a pathogen, the specific immune system will not work. The second time you come in contact with it, it will. First time you come in contact with a pathogen, you're either going to die, or the non-specific immune system is going to be able to overwhelm that pathogen and successfully defend your body against that pathogen. You may have a really terrible case of that particular disease, but you didn't die, and that's all that matters. If you ever come in contact with that pathogen again, the second time you come in contact with it, this immune system is going to come into play the exact same way it did the first time. But the second time, your troops, these guys, have already been trained on how to attack that pathogen. So this time, the specific immune system is going to come into play as well. So you've got two branches of the military coming into play instead of just one. And sometimes these guys are so good at their job, they can stop the pathogen from the get-go from even causing you to get sick. And if you do get sick, it's probably going to be a significantly reduced infection. You're probably going to not get nearly as sick because you've got two parts of the immune system. You'll have a better chance of getting better, of recovering, versus just this particular part of the immune system. So that's the catch here. The first time you see a pathogen, this guy is going to come into play. This one cannot work the first time. It only works the second time. You have to see the pathogen the first time for this thing to come into play. The second time is the only time it comes into play, and thereafter, not the first. So the innate immune system is going to include things, like I mentioned, that you're born with and things that you will have throughout your entire life. It's going to include things like your skin, things that you don't really think about, physical things that keep bacteria from getting into your body, fungus cells from getting into your body, spores, things like that, stuff that keeps bacteria and microbes out of your body. Skin's a very obvious one. It's a big giant organ. It's covered in lots of oils. It's got a slightly acidic pH. Bacteria don't like that. It's covered in salt. It dries bacteria out. They don't like that. It's too thick. It's too solid. There's not a lot of breaks in the skin. That's why if you have a wound, you're more likely to get sick. Intact, unbroken skin is a very, very, very difficult thing for bacteria to get through. They don't burrow through that so well. So it works very well to keep bacteria out of the muscles and things underneath it. They would have a very easy time causing infection in. Mucus and mucous membranes. Mucus is a really good way to keep things out of your body. Now, what's a mucous membrane? So mucous membranes are the wet parts of your body. Things like the urinary tract, the reproductive tracts, things like that, the anus, the ears, the eyes, the mouth, anything that's wet, kind of things like that. So mucus, what it does, all that's the wet part. What that's there for is it's to trap things inside of it. So mucus, essentially how a booger forms, is all the mucus inside of your nose is meant to trap things that you inhale. So all the little hairs inside of your nose and stuff will trap fungal spores and bacteria and things that you inhale up your nose. And the mucus will then coat them all up and blob over them to trap them there. So the hairs catch them, the mucus traps them in place, it sticks them to it. And then eventually the big blob of mucus builds up, and that's a booger. So if you didn't have the little hairs or the mucus to keep all that stuff in place, you would just inhale all of those mold spores and all of that bacteria all day long, and it would go straight into your lungs. And that would not be good. That would be a very easy way to get sick. So mucus and mucous membranes, all that mucus is literally there to keep stuff out, keep bacteria from having a difficult time to sticking to the surface, that adhesion concept of causing disease. So that's pretty much what they're there for. A lot of the mucus is also going to contain some enzymes and things that we'll talk about in a little while to kill bacteria, prevent them from growing. And then one that's kind of odd is the flushing of the urinary tract. So the flow of urine can push bacteria and other little organisms out of their urethra. So it's kind of like the concept of a fire hose. So the water's being pushed through, the urine's being pushed through so fast, those guys are so tiny that it's like getting hit by a fire hose as the urine passes through the urinary tract out the urethra. Males have a longer urethra than females, which is why they are less likely to get urinary tract infections. Also has to do with the location of the urethra in proximity to the anus, the bacteria that are more likely to cause UTIs. So anyway, your body also has some physiological defenses, things like fevers, things like inflammation and things. A fever is going to help raise the body's temperature to help increase the activity of the immune system, increase movement of the immune system to a particular area. Inflammation is also going to help widen the blood vessels and things, which we'll talk about in a second, to help parts of the immune system cells travel faster through the blood vessels to the location of the inflammation, the location of the infection. So let's talk about the very first line defenses, the very first thing that a bacteria that wants to invade your body to make you sick has to deal with to get inside of your body. These are called first line defenses. So here they are down here, what they are and how these particular things are going to keep a bacteria out, to keep microbes out of your body. So if an organism wants to make you sick, or it's going to try to get you sick, it has to overcome these first lines of defenses first. If it can't get inside of your body, it cannot make you sick. So this is what it has to do. It has to overcome one of these first lines of defenses to get inside of you. And all these things are meant to keep them out. It has to get inside one way or another. You have lots of ways that are meant to keep them out. So the very first one, like I mentioned, is your skin. Big, giant, hard, rigid, salty, dry, slightly acidic, very, very, very solid, very thick tissue barrier to keep bacteria out of your body. All this moist, warm muscle underneath is easy pickings for bacteria. They would love to get on top of that, but they can't because the skin keeps them out. All the stuff inside of your skin, all those oils and things, the bacteria don't like it. All the salt's going to dry them out. It's slightly acidic, so they don't like that either. So all the stuff on your skin is going to work to actively kill and prevent bacteria from growing on your skin. So once again, the skin itself is very thick, so bacteria cannot burrow through it. Now, if you break the skin, if you cut the skin, or you get a wound or a piercing or a tattoo or something like that that breaks this first line tissue barrier, you expose that underneath tissue area that's not meant to keep bacteria out. It doesn't need to keep bacteria out because bacteria aren't supposed to be there. So if bacteria do get there, you cut the wound, you cut yourself or something like that, these guys don't have any, this area doesn't have any way to keep bacteria out, any defenses for it quite as much. So it's easier for them to set up a shop and cause an infection down here if they can get in through the tissue barrier. So this is why it's very important to keep your skin healthy, keep it moisturized and things like that. Broken skin, if your skin is very dry, it's easy for bacteria to get down inside of the cracks, to get down in here and things like that. If your skin's cut, that's why they'd say wear gloves and band-aids and things just to keep organisms out of those wounds and things like that, because your first line of defense has been broken. Now they can easily get through the front door. Essentially, this is like having your house. Then having a wound is essentially like having your house and then leaving the front door open. Kind of defeats the purpose of having all the walls around you. So once again, you have lots of other things inside of your skin, sweat, lots of other acidic things that keep bacteria from growing on your skin. Saliva, tears, mucous membranes also have tons of enzymes, things like lysozyme, that are meant to break down the sugars that are found on bacteria cell walls. And you can test this at home. It's a very interesting little trick. You can take a piece of white bread, just plain old white bread, a little square, put it on your tongue, and let it sit there for a couple of minutes, and eventually it will start to taste sweet. And that's the lysozyme in your saliva that's breaking down the sugars inside of that bread. It does the same thing to the cell walls of the bacteria. It breaks it down. And that's meant to keep bacteria out of your mouth. That's not supposed to be there. So if a bacteria and things are living on your skin, commensalistic organisms, we've talked about these a little bit, if they're living in your mouth and things like that, they are supposed to be there. They have evolved to survive in a very highly acidic environment, like your skin, a highly salty environment. They are meant to be there. If a bacteria is not supposed to be there, the saliva, the enzymes in the saliva, the salty environment should kill them, and that's the point. That's how this stuff is supposed to work. If a bacteria lives there, it's not supposed to cause you problems. And all those good bacteria that live there, I mentioned this concept earlier, the good bacteria crowd out the bad bacteria. There's no space for the bad bacteria to live on your skin. If they land on you, all the good bacteria on your skin are taking up space. All the staph, all the stuff on your skin, or the Corynebacterium, are taking up all the good occupiable space. They're eating all the food. So if a bad pathogen lands on your skin, there's no food for it. There's no space for it to live. So it's going to be crowded out by the good bacteria. So that's why you want to keep your skin healthy as well, to keep the bacteria that live on your skin healthy too. They play a very important role in your immune system. So once an organism can attach to your tissue barrier, if it can overcome the tissue barrier, if it can get to the skin and firmly cling a hold of it somehow and survive there, then it's going to start to be able to cause infection. That's the very first step. You have to be able to get to and hold on to the tissue barriers. Most of them can't even survive onto the tissue barrier in the first place, the bad pathogens, if you're not supposed to be there. But if you can and you can hold on to it, then you can set up shop. Like I mentioned, there are quite a few things that are found in your saliva, in your tears and things like that are meant to break down sugars inside of bacterial cell walls. Once again, they're going to break down the peptidoglycan, the sugars inside that cell wall, so they're more effective on gram-positive bacteria. And this also makes a little more sense if you think about it in detail. Gram-positive bacteria are a little more likely to be found in the environment. They don't dry out as much because of that big, thick peptidoglycan layer. Gram-negative bacteria tend to be found inside of living organisms a little more often. So the chances of coming in contact with a gram-positive in the environment is significantly higher than a gram-negative. So chances of gram-positives falling in your eyes or getting in your mouth and things where you're going to need the ability to break down that peptidoglycan, that exposed peptidoglycan cell wall, is very high, whereas a gram-negative getting in your mouth is fairly low, and you don't need, comparatively anyway, and you don't need to be able to break down peptidoglycan because they have a hidden peptidoglycan layer. You can't get to it, kind of thing. So, as well as just the simple immune system that we're going to talk about in just a second, all of the cells, things that you guys are familiar with, lymphocytes, white blood cells, and things like that, leukocytes, we're going to talk a little bit about the lymph system, just a little tiny bit. And the lymph system runs along with the traditional circulatory system. The circulatory system is all of the blood vessels and arteries and things in your body, the heart, that moves the blood around. And that's going to be what your immune system lives in. All of the cells that we'll talk about in a second live inside of the circulatory system for the most part. They're going to live inside the blood vessels. That's how they're going to move around. The blood vessels are the road for your immune system. All the cells move along the inside of the blood vessels from place to place. So along with that, you're going to have the lymph system that travels right around pretty much the same place. You can see the lymph system over here, the circulatory system over here. It pretty much goes right along the same place. Now the lymph system, it goes down into right in here. The lymph system and the circulatory system are going to overlap a little bit. And the circulatory system is going to have cellular debris that the immune system's killed, old cells, dead cells, just garbage, trash. Floating around inside of it from the destruction of pathogens, cells dying over time, all kinds of junk floating around inside of there. So the lymph system is going to clean it out. The lymph system is going to sweep and clean out all of that garbage and that junk from the circulatory system. It's going to clean out all the garbage. One of the other things that it's going to do is it's going to make sure that none of that garbage is bad. If it finds a pathogen inside of that garbage, something that could potentially cause harm to the living organism, the human as a whole, it's going to take that pathogen piece and back to the nearest lymph node and kind of alert the body. Oh, hey, look, this particular pathogen has been found in the body. I'm going to hang it up essentially on the nearest lymph node, the wall of the nearest lymph node, so the rest of the immune system can see it. And then the immune system is going to travel via the circulatory system. A bunch of white blood cells and things like that are going to travel via the circulatory system to that lymph node and set up shop there. And that's where they're going to stage to go fight the infection. So that's why your lymph node swells up. The lymph system has found a problem. It's brought it back to the nearest lymph node to tell the immune system, hey, this is what you need to look out for. We have a problem. All the cells of the immune system, a ton of them have migrated to that lymph node. They're now waiting to be sent out to battle the infection in your arm or something like that. So let's go ahead and talk about the cells that are associated with the innate immune system. So we're going to go ahead and start this off with just the innate immune system, and we'll finish it up with the adaptive immune system. So these are the cells of the innate immune system. These cells are going to have their exact same response every single time, regardless of what type of pathogen they come in contact with, even if it's not a living cell. It could be a splinter. So these are essentially considered blood cells. When you think of blood cells, you think of red blood cells, white blood cells, and platelets. So if you're a red blood cell, a white blood cell, or a platelet, red blood cells carry oxygen, erythrocytes. White blood cells, leukocytes, are immune cells, and then platelets, thrombocytes, clotting cells, and things like that. So we're going to be mostly talking about white blood cells. It's pretty much the only thing we're going to talk about. Regardless, though, of which type of cell you're going to be, you are all going to begin your life as something called a hematopoietic stem cell. Stem cells are cells, baby cells essentially, that really don't have a job yet. They are essentially just little cells that have not been exposed to chemicals called colony-stimulating factors. And these colony-stimulating factors tell the cells what to do. So this is essentially a baby cell that could become a liver cell or an eye cell or things like that, whatever. And a colony-stimulating factor is going to tell it what to become. Now, in this case, a hematopoietic stem cell is a cell that's destined to become a blood cell. It could become one of these three different types. There's quite a few different types of leukocytes. So a hematopoietic stem cell is just a baby blood cell that hasn't been programmed to become anything yet. It could become one of countless different types of blood cells, and we'll talk about some of them here in a little while. So regardless of what type of blood cell you eventually become, you all begin your life as something called a hematopoietic stem cell. So this is your hematopoietic stem cell. Hematopoietic means blood. So they're all going to start their life inside of the bones. So we have bone marrow down here inside of your long bones, femurs, and things like that, where the vast majority of your immune system is going to be made. They're all going to be birthed and made, if you like, inside of your bone marrow. So all those little hematopoietic stem cells are going to be birthed inside of the bone marrow. And then they're going to be exposed to a colony-stimulating factor. And we'll talk about what that is in a little while. It's essentially a chemical that tells them, a hormone that tells this cell, hey, this is your job now. This is what you need to become. So once they are exposed to that, they will take a different path depending on the different type of chemical that they're exposed to become a different type of cell needed for the immune system or to carry oxygen or whatever. So we're going to start with these guys over here, the innate immune system, and we'll finish up with the adaptive immune system. So leukocytes, these are the cells that are associated with the innate immune system that are actually dealing with killing and preventing infection. So leukocytes, leuko just means white, cyte means cells, so white cells, pretty easy on that one. These are the cells that are going to kill bacteria, kill microbes, kill mold spores, kill parasites, pretty much anything that's not supposed to be inside of your body, they will destroy it. There are quite a few different types of leukocytes. They're down here, and then we'll talk about them each individually in just a second. So let's start with granulocytes. You can see here there's a couple different types of granulocytes, and we'll talk about them in just a second. So granulocytes get their name because they contain something called granules inside of them. Let me flip back here. So you can see the little granules down here inside of our granulocytes. So granules are essentially just little tiny water balloons of digestive enzymes. So the granulocytes are going to phagocytize. They're going to eat a pathogen, a microbe, or something that's bad. They're going to phagocytize it, bring it inside of their cell. And once that organism has been brought inside of the cell, they're going to throw those granules at it. They're going to burst the granules on the surface of that pathogen. The digestive enzymes, in theory, will break down that particular organism and destroy it. And that's what a granulocyte does. That's how they get their name, from those little granules of digestive enzymes inside of them. And now the problem with granulocytes, same with every single concept of a water balloon fight, is when you run out of water balloons, the fun's over. And that's what happens to these guys. When they run out of granules, they die. And they can't get new granules. So these guys have a limited lifespan. They're formed with a certain number of granules. They're birthed, if you like. They don't get any more granules, and once they use up all their granules, their lifespan is over. They do not replenish any more granules. So the most commonly known type or thought of type of granulocytes are neutrophils. So cells, these different types of cells, neutrophils, eosinophils, and basophils, get their names from the type of dye that's used to see them. A long time ago, they would make a blood smear where they would just take a sample of your blood, put it on a slide, and then they would pour different types of stain on it. Different types of stain would stick to different types of blood cells, and the type of stain is what gave them their names. So neutrophils didn't stain very well. They were neutrally staining, hence the name. This is a neutrophil down here. Um, these guys are essentially the most phagocytic cell. They're going to eat anything they can get their mouths, anything inside they can get, anything they can get inside of their mouth. They will try to eat and digest with their granules. They will bring it inside, phagocytize it in, and destroy it. So this is a red blood cell, a little thrombocyte here, platelet, and our white blood cell here. Mature neutrophils are known as polymorphonuclear neutrophils. Poly meaning many, morpho meaning shape, nuclear meaning nucleus. So if you have to identify these things, which you will at some point in time for nursing school or medical school or vet school or whatever, this is what they look like. So this is the nucleus here. It's got different, it looks like it's got multiple nucleuses. It doesn't. You can see the little connectors there. And that's how they get their name, because it looks like they've got multiple shaped nucleuses, but they don't. So polymorphonucleocytes make up a little more than half of all the white blood cells that are floating around inside of your body at any given time. They're just going to be chilling out inside of the bloodstream. That's where they live, just waiting for something to happen. If you get infected or you become infected, you get a microbial infection of some sort, the numbers of PMNs in your body are going to instantaneously increase. You have about 100 more of them or so just waiting and ready inside of the bone marrow, just ready to be called into action if they are needed. So the immune system recognizes that you have a problem. It calls into action the neutrophils that are waiting inside of the bone marrow, and they come out and start fighting against that pathogen. So one of the things that these guys do and how they kill things is they gather in a big, large area, and they start digesting things in mass. So when they start digesting things in mass, they're going to phagocytize them. They're going to use their granules up. When they use their granules up, they're going to die. And when they die, their little bodies stick together in a little goopy mass of nastiness that's proteins and little dead cells and bacterial grossness and cell remains, all kinds of nastiness. And it's going to pile up on top of itself and create something called pus. So if you get a pimple, this is exactly what's going on there. You get a clogged pore. The bacteria inside of that pore are causing a problem. The neutrophils gather in that area to start killing the bacteria. As the neutrophils die, the little bodies and things start to pile up. And then that's what causes the pimple. And when you squeeze the pimple, all the stuff that comes out is all their little bodies and all the little cell junk and all the digested extracellular nastiness in the form of pus. The other thing that this pus does is it's a giant sticky grossness blob that traps bacteria to it. So it kind of has a purpose. It's not only just their little bodies, but their bodies are sticky, and anything that comes in contact with it will also stick there too. So if bacteria are there, the pus is going to stick them to it. Any digestive enzymes that may have been released from the dead cells or that from busted granules that got out or whatever that were not used, all those digestive enzymes and things will hold inside of that little pus ball, that little pus net. So the bacteria that are stuck inside will not be able to do any harm, and all the enzymes will still be able to function instead of being just kind of lost to the body. So that's what the point of pus is. So neutrophils are terminal cells. Once they become a polymorphonuclear neutrophil, they don't reproduce anymore. They are that, and once they are done with their granulocytes, they are done. They are one of the most highly phagocytic cells in our body, and pretty much the main source of immune system fighting that goes on for just your day-to-day bacterial infections, just normal things that your body comes in contact with. You see these all the time if you squeeze pimples or you get a boil or little abscess or things like that. This is pus. That's why it's white. White blood cells kind of things is what you're looking at. And these guys are almost exclusively found inside of the bloodstream, except for during the process of active infection on the skin or things like that when you're getting inflamed in an area. So if you get a cut on your skin or something, these guys will migrate through the bloodstream to the area of infection and then migrate through the tissues out of the bloodstream up to the skin to help fight the infection. And that's how they get there. You have blood vessels that run very close to the surface of the skin, so they'll just take the little extra distance to the surface of the skin. Very easy for them to do so, but they very rarely leave the bloodstream unless they have a reason to. So the next type is called a basophil. These guys stain with basic pH dyes, anything 7 or higher, which is why they get their name. And they come out dark purple for the most part when you see them. These guys are full granulocytes, and their granulocytes are going to contain digestive enzymes as well as two very important things called histamine and serotonin. Serotonin makes you feel a little better, so you're less likely to want to sit around and be miserable all day, so you'll get up and hopefully do a little more exercise. And histamine contains, or histamine is an inflammatory agent for your bloodstream, a vasodilator. It causes your blood vessels to open up. And the easiest way to explain this is when you have an infection in your arm, so let's say in your hand, your immune system has to travel through your bloodstream, your blood vessels and things to get to that part of your hand to help combat the infection. Now, your blood vessels are quite small. Just a certain amount of the bloodstream or the immune system can travel through there at one time. You have such a limited space, and a limited amount of cells can go through there. Histamine causes the blood vessels to dilate, to increase their size, to open up, which allows more blood to go through, which increases the heat, which causes the enzymes that are in that blood cells that are the immune cells that are already there to work faster, to work a little better. The increased blood flow is going to help carry away pathogens and things like that, help carry away cellular debris to the rest of the body to help to get away from that disease site. It's also going to allow more of the immune system to travel through that widened, that dilated blood vessel. So histamine does a really good thing for your immune system to help fight off infections. So if you get an allergy infection, antihistamines narrow down the blood vessels and keep that from happening. So your face doesn't run off and your eyes running off and your nose running off and things from your allergy infections, because that's what's happening with histamine. It causes all your blood vessels and things in your body to dilate. So you get all runny because of allergies and things like that. So we'll talk about what a mast cell is in a second. But a basophil and a mast cell are essentially the exact same thing. They do the exact same thing. These guys phagocytize something. They bring it inside their cells. They throw all their granulocytes at it. Their granules contain histamine and serotonin, which once the cell is done, all that serotonin and histamine is released into the body, which contributes to the inflammation response. Basophils live inside the bloodstream. Mast cells are the exact same thing. They just live inside the tissues, and that's the difference. They look the same. Literally, the difference is they found in the tissues, whereas basophils are found in the bloodstream. It's pretty much the only difference. Eosinophils, they're stained by the dye eosin. You can see them down here. Eosin is this really pretty kind of magenta color. These guys are responsible for allergic reactions. Their essential role is to fight off parasitic infections. Humans don't get parasitic infections very often anymore. We used to get them quite often back in the day, but not so much anymore. And these guys, what happens is they get bored. And when they get bored, their numbers start to increase. And what would happen a long time ago, you had a parasitic infection. These guys would have something to do. They'd fight off that parasitic infection. Their numbers would go down, 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 down. You get another parasitic infection, their numbers start to increase. You had a parasitic infection, they had something to do, and so on and so forth. The cycle just repeated itself over and over and over again. But nowadays we don't, and these guys get bored. They start to divide. Their numbers get really high, and they don't have anything to do. And then something that's floating around inside of your bloodstream at the time in really high numbers that's not supposed to be there, they will attack, whatever it happens to be. If it's latex or pollen or anything like that, just whatever it happens to be that they come in contact with when they're bored, they are going to trigger as, oh, that is what you are now allergic to. They are going to trigger an allergic response to that. They're going to teach your body that this is now what we are allergic to. And that's how allergies work. These guys get bored and they attack some random thing inside of your bloodstream at the time that happened to be there. This also explains why allergies can go over time, come and go. These guys have a limited lifespan. They will die over a certain time period. If you don't re-expose yourself to that allergen, the chances of these guys being able to survive for 30 years and continue to know that you're allergic to it are pretty slim. So the ones that are replacing these guys won't remember what they're allergic to. They've never seen it. So they're not going to really so much worry about it. If you keep re-exposing yourself to that allergen over and over again, you eat peanuts every 6 weeks or something, you're going to keep refreshing the cells so they will remember it, kind of thing. So you can grow out of allergies, and this is why. There are people out there that purposefully infect themselves with pinworms. We talked about pinworms in a previous section of this lecture course. That purposefully infect themselves with pinworms so they can keep their allergies in check. It's very easy to get rid of pinworms, so that's why they do that. They keep a pinworm infection for a couple of months. It teaches their immune system to not be bored, give it something to do, and then they don't have any allergies for a while. Supposedly it works. We'll talk about phagocytes for a little while. These guys start out as a monocyte, a mononuclear phagocyte kind of thing, and these guys will eventually turn into either one of a macrophage or a dendritic cell. And we'll talk about what these are in just a second, what a dendritic cell and a macrophage are. So macrophages are pretty cool. Macrophages are a type of the immune system cell that plays a role in both the innate and the adaptive immune system. And we'll talk about what they do in both in just a second. But in the innate immune system, what they do is they are essentially the high performance janitors of the body. They are very highly phagocytic. They will eat anything they can get their little mouths around, bacteria, mold spores, dirt, dust, anything they can get their little mouths around, they will digest and eat. The really cool part about these guys is they are able to migrate out of the body, out of the bloodstream, and live in the tissues. Most immune cells cannot do this. So these guys can live in the lungs, which is really cool. This helps defend your body from things that you inhale all day long. So these guys don't need the blood system to live. They can live inside of your lungs. They can live in mucous membranes. They live all over the body, which is really cool. So they can just help fight infections. And these guys kind of work as the kind of the guards that look for problems. Since they're everywhere in the body, these guys are going to be one of the first cells that picks up that there's a problem if a pathogen comes in. These guys will find it first. They can live for a really, really, really long time, a couple of years at the most in certain locations. So this is really cool. And the other thing is they are not terminal. Means they don't die. They can also turn back into other different types of white blood cells if they're needed. So if the immune system runs out of a certain type of cell, runs out of basophils or something, it can release a new chemical, a new colony stimulating factor that triggers a bunch of macrophages to turn in to eosinophils or turn into basophils or whatever is needed, and that's pretty cool. So the different locations for macrophages throughout the body gives them different names, but they're all the same idea. They all function the same, and they're essentially all macrophages. So like I mentioned, they travel around from tissue to tissue. They can move around within the body, and they just clean up stuff that you see every single day. They're kind of just the street cops. They just take care of normal day-to-day cleaning, the mold spores, the dust spores, the bacteria stuff that you inhale, just stuff. They're capable of making brand new granulocytes for themselves, which is cool. They're the only cells that can do that. They don't run out of lysozyme. They don't run out of enzymes, which is pretty neat. And you can find them everywhere you look throughout the body, which is pretty cool. So once they revert back, if they do, to a different type of cell, they cannot become a macrophage again. They're stuck at whatever they become back into, and then eventually they will die when they run out of granulocytes as that other cell type. So one of the cool things about macrophages is if they can't phagocytize, they can't eat and destroy whatever it is by bringing it inside of their cell and throwing their granulocytes at it. If they can't destroy it that way, they adopted the football strategy of dogpile. So there's some little mold spore, bacteria, some little pathogen, something in here that the macrophages were, see, all these little dots are macrophages, by the way, that the macrophages were unable to eat. They couldn't digest it. So they just dogpiled it. And the idea here is that they're either going to choke it from nutrients, choke it from oxygen, or keep it from being able to reproduce. So even if you don't starve to death, you're not going to be able to do anything. So that's kind of the idea. They just wall it off from the rest of the body, and they entrap it in here so it cannot cause problems. And this is called a giant cell. And once this organism dies, they will eventually be able to digest it, which is kind of cool. And once again, I mentioned that they play a really big role in the specific immune system since they're out in the body and every single tissue throughout the body. They can pretty much sense if something's wrong. They pick up a pathogen first. They're kind of one of the um, first things to go, oh, there's a problem. Let me raise the alarm. So dendritic cells are another type of cell that plays an alarm role, a sentinel cell. Is another term for these. And these guys live inside of the lymphatic system. These guys live in the lymph tubes. Instead of the circulatory system, they're going to live in the lymph system. And what they do is they stretch out like a spider web throughout the lymph system. So I mentioned earlier that the lymph system is going to clean the circulatory system. It's going to clean out all the junk inside of it. And all the junk that's swept through the lymphatic system before it's permanently cleaned and destroyed and gotten rid of is going to be washed through the tube of the lymphatic system, which has a dendritic cell, a spider web, stretched out all the way across it. So you can see the tube behind it where the fluid would go through, and this dendritic cell is stretched out through the opening of that hole. You can see the little bacteria and things that are stuck in the neighboring sentinel cells. So what's going to happen is that little sentinel cell is going to identify if there's a good bacteria or bad bacteria, pathogens, and things like that. If it identifies a pathogen, it's going to alert the rest of the immune system. So if this guy gets a bad thing stuck in his little spider web, he's going to raise the alarm for the rest of the immune system. And we'll talk about these guys in the adaptive immune system, but they're going to be how cells kind of talk to one another a little bit. So the complement cascade is part of the innate immune system as well. This is not involving cells. This is a very interesting metabolic pathway that's essentially a really long convoluted set of steps and enzyme reactions and binding to cells and surfaces of cells and things to poke holes inside of the cell membranes and cell walls of bacteria. You poke a hole in the surface of a bacteria, the cell contents are going to leak out, and in theory it will die, and that's kind of the purpose of this. So this is something called the complement cascade. You have to have a particular antibody or bind to the surface of a pathogen, or a particular type of cell will bond to the pathogen, which initiates this complement cascade to trigger. Really long complicated way to say it pokes holes in the walls of bacteria. Now, unfortunately, it will try to poke holes in the walls of every single invader that it sees. This is the innate immune system. It cannot tell the difference between things that it sees. It tries to attack every single thing that it comes in contact with the exact same way. So it'll try to poke holes in bacteria and viruses and mold spores and splinters, everything that it will come in contact with. And if it's a living cell, it will poke a hole, and that particular organism will no longer be able to sustain osmotic pressure, and it will die. So how does the body talk to the innate immune system? How does it tell the innate immune system what to do? So our colony stimulating factors are looped into this, which we talked about a second. We'll talk about a couple of the other chemicals and things that are used for the body to talk to one another. So essentially what these are, they're tiny little proteins that the brain is going to release, kind of like hormones and things like that, that direct different cells, direct groups of cells, direct parts of the immune system to do different things. And this is how the body is going to direct different parts of the cells, different parts of the immune system to do different things. So one of the ways that the immune system is going to tell different cells and things what to do is a chemical called interferon. Now, interferon is going to be used primarily to fight viruses. There's 3 different forms of interferon, A, B, and alpha, beta, gamma, and it's used to essentially tell cells you're going to die. What's going to happen is cells that are infected with viruses, they don't really recognize that they have a problem. When they become infected with a virus, if you recall, viruses want to replicate their own DNA, their own RNA, and make new copies of themselves. So there's going to be a large number of double-stranded RNA that will show up inside of a virally infected cell. Once that cell recognizes that it has become infected, interferon will start being produced throughout the body. And what's going to happen is once interferon's released, it's going to bind to cells that are having problems. And it's going to stop them from being able to reproduce. It's going to stop that cell from being able to do anything at all. So the viruses that are found inside of it will eventually die. It ceases the cell's reproduction. And it ceases the cell's ability to make more proteins, which means the virus that's contained inside of that cell will no longer be able to be reproduced. Now, the problem with that is, since the host cell is had all of its proteins production turned off, it will die as well. Essentially, once interferon is bound to a cell, the immune system will damn that cell's life. It will send it into oblivion. It is virally infected. It is no longer good to have around. It is dangerous to us. So once interferon has been bound to that cell, turned off the functionality of the proteins, that cell's life is already signed over. It is done. So interferon can be really useful for treating viral infections in a lab. You can only give it for a couple of days, though. Either you're going to get better because that viral infection is going to burn itself out, or your immune system is going to take over, or you're going to give so much interferon that you're going to kill the person that you're trying to help. Enough cells will be infected that interferon will eventually start killing important cells. So your body only naturally produces interferon for a couple of days. You could give it by intravenously, in theory, forever, but eventually it will start killing your host, your patient. Initially, it was thought that you could give interferon from patients from different species to humans and different species to each other. But it's actually comes to find out that species-specific interferon is a pretty common thing. It's very hard for us to take other species interferon. And that makes sense if you think about it. If you refer back to the idea of the host range in viruses, it would make sense that we have viruses that are unique to humans, so therefore we have interferons that are only unique to human viruses, since those viruses aren't found in any other species. So now we use bacteria and yeast to make human interferon in a lab that they purify away, and that's what they use in a hospital setting. So interleukins. There's a bunch of different types of interleukins, and essentially what they're going to do is they're going to contribute to the inflammation response. They're going to tell the body, hey, it's time to raise the temperature to cause a fever. They're going to say, hey, it's time to cause vasodilation to release the basophils. It's going to trigger the body's immune response to trigger the body to get ready to fight off an infection. Colony stimulating factors. I've talked about these already. These are what are going to trigger those different types of blood cells, that hematopoietic stem cell, to become something else other than what it is. It becomes exposed to a different type of colony stimulating factor. There's a couple different types of them. You can see them down here, GM and IMS. Depending on which type you're exposed to and the concentration of it, you will become one of a different type of white blood cell, which is pretty neat. And the body can just release different types or different amounts of colony stimulating factors at different time, which will be used and regulated to determine what the body needs at the time. So if you need to fight off an infection, or you need to replace these in the body, you can grow more of whatever you need at the time, which is pretty neat. Tumor necrosis factor is going to trigger cells to kill themselves. This is programmed cell death, apoptosis. Essentially what happens is the cell destroys its own nucleus and then packages away all of the important cellular components, destroys those as well, and then self-destructs. This is going to read a cell and determine if the cells have problems, if they have mutations in them, if they're growing correctly and things like that. And if it determines if there's a problem, it will trigger that cell to direct itself into apoptosis, direct itself to die, because it doesn't want to continue that damaged cell reproducing and causing problems for the host. If you have problems, or if you have a mutation in your tumor necrosis factor genes, it can lead to a predisposition to developing cancer, because your body's not quite as good as reading the cells as someone who has a fully functioning tumor necrosis factor gene. Chemokines, if you recall from our earlier lecture about how bacteria move around, we have chemo means chemical, kine, kinesis means movement kind of thing. So chemokines are chemicals that are used to talk to the body, that are pretty much how it moves from place to place. So chemokines are going to be used kind of the exact same way as the idea of positive and chemotaxis. Is what the word I was looking for. So apply this in the same idea. An organism has an infection on its cells in its arm. Now, when a cell is damaged, it's going to release chemokines, and chemokines are essentially "help, I'm dying, come help, come help." Essentially, those kind of messages, little sensors that say we're damaged, we need help. So if you recall our positive chemotaxis, you walk in the door, you smell some cookies, and you follow the cookies back from the source of least amount of smell to the source of most amount of smell in the oven. And this is how this works. There's a lot of cell damage up here, a lot of chemokines. As it's dispersed through the body, through the bloodstream, it's going to become less and less and less and less the farther away you get from it. The immune cells will follow that chemokine. That's what it's meant for. It's meant to attract other immune cells back to the location of the infection. So the other immune cells that are all throughout the body will then follow that least concentration all the way back to its higher concentration, all the way back to its original source to help fight off a source of infection over here. The reason for that is keep in mind that all these cells work together for the good of you. They're all together because if you die, they die too. So they don't want to die and they don't want you to die. Essentially, keep in mind they don't think. It's just kind of how they're programmed to work. So they need to go back to one area to keep the organism as a whole healthy. So this is what chemokines are for. They're meant to bring the organism's immune cells to a particular location. So this is the concept of inflammation. Very, very, very simple. I've talked about the inflammation response, how it's triggered a little bit. So you break the skin, that first line of tissue barrier, it's broken, the organism pathogen gets inside through that broken tissue barrier. The macrophages in the body, or the mast cells, which are the exact same thing as basophils, will then detect that pathogen in the body. Once they've detected that pathogen in the body, they will release their chemical sensors that tell the body, hey, we have a problem. We have detected a pathogen. These chemical sensors will then travel into the bloodstream, which will then trigger the basophils and everything else to dilate the blood vessels. So the blood vessels, you can see here, they become dilated, which allows more of the immune system to travel through. This increased blood flow is going to carry more of the cellular debris away, the nastiness, dead cells, and things that will help wash that stuff away. It's also going, like I said, the main point is once again just to allow more of the immune system to travel through it. So those chemokines will attract back the immune system cells to the source. They will travel from wherever they're at to the particular location of the infection. And this is a process called diapedesis, where they squeeze through the bloodstream, out of the bloodstream into the tissues. So the white blood cells will squeeze through, diapedese through the cells of the bloodstream into this tissue of the body tissue. Once they've entered into the body tissue, they will start phagocytizing up all of the pathogen in the area. They're using up their little granulocytes. They will eventually die. Once they die, the immune system from the body will just destroy them and get rid of them. It's just cellular leftover debris. And the wound heals up, and they destroy the pathogen on the inside as well. So this is essentially the concept of inflammation, how it should work, and how, in theory, this should play out. Is these guys should eventually eat all the infection, knock it down to the point where it's no longer causing a problem, and the body can just handle it, and the immune system will wipe it all up. And then the wound will become healed and keep everything out. The immune system response for this, it is part of the innate immune system, is going to be the exact same if you get a splinter, if you get a bacterial infection, if you burn yourself, it does not matter. All these all the white blood cells are going to rush to that area regardless, thinking that you have an infection, because you also might have broken that tissue barrier, so that's why they're going to rush there. The exact same response no matter what it is. It's that concept of chemotaxis again, that going from low concentration of chemicals to high concentration of chemicals. So phagocytosis, we've talked about that a lot, and this is how this works. This is the word form of that, but this is phagocytosis. A macrophage or another type of cell is going to bring inside a pathogen. In this case, it's a virus. They're going to bring it in. You can see that. They're going to bind their digestive granules with it. This is called a lysosome or phagosome. Sorry, a phagosome, not a lysosome, a phagosome. They're going to bind their little digestive enzymes with it. The little digestive enzymes will break down whatever's inside of the phagosome, hopefully killing the pathogen. And then they will just release the waste material out, and then that will just be wiped up with the rest of the immune system and destroyed. Now, I've talked about bacterial capsules and things, how they avoid the immune system, and this is what that does. A bacterial capsid, so when the lysosome, little digestive enzyme, bind, the capsule protects them from being destroyed, so the digestive enzyme doesn't do anything to them. So there are a couple of different factors that can influence the overall immune response. Age, as you get older, the ability for your immune cells to grow and reproduce and work as well as reduced. Your overall health, if you don't eat a good diet, if you don't exercise, once again, that makes sense. Iron levels have quite a lot to do with the health of your immune system and your ability to carry oxygen around inside of your body. If you have a fever, immune health, and there are a bunch of others that you could probably think of as well. So let's go ahead and start the specific immune system. This one's pretty easy to understand. The main players in this one is an antibody as compared to cells in the adaptive immune system. So a little bit of history of this one, how it was discovered. If you've ever seen the movie Balto, this is essentially the story of Balto. So this guy right here, Frederick Loeffler, he worked with a bacteria called diphtheria, Corynebacterium diphtheriae, and this is it up here. It looks kind of like a little club. It's a polymorphic bacteria. It does not have a shape. Pleomorphic bacteria. There's no set shape for these guys. Kind of a weird little shaped bacteria. They're often called club shape. Well, anyway, the symptoms of diphtheria. You get this organism inside of your body, and it lives inside of this little membrane in the back of the throat. And this is kind of one of the most common diagnostics of this. This is a little pus ball. The little diphtheria organism lives inside of the little membrane here. And it can produce toxins that can travel throughout the rest of the body, which can cause problems with other organs and things like that. One of the main ways of death that this organism causes is this little membrane here will break off and fall down the throat of the person that it's infected. Now, in adults, that's not a huge deal. If this thing falls down your throat while you're sleeping or while you're awake, you're going to cough, and someone can either help you or hopefully you'll cough it out. Now, in babies and infants and young children, if this thing falls down their throat, it breaks off while they're sleeping. They don't wake up. They just suffocate. I mean, that's an unfortunate reality of life. So this is how this stuff kills you. And sometimes you can just you don't even have to have the little membrane here. And the toxins that this organism produces is enough to kill you as well. So it just kind of depends. Well, this guy right here, Frederick Loeffler, was a German scientist, and he looked at this organism, diphtheria, and he wanted to figure out how it was causing problems in the rest of the body, but it was only found in those little throat membranes. Now, he didn't know that it produced a toxin. He didn't know any of this stuff yet. He was just trying to figure all this stuff out. So he figured that he kind of guessed that it was probably producing some sort of toxin, some sort of something in the membrane in the throat that was being spread throughout the rest of the body. So he couldn't find that bacteria anywhere else inside of the person's body except for in their throat. Everywhere he looked in the bloodstream, he couldn't find it anywhere at all. Any other organs, no tissues, nothing. But that organism was causing problems in other organs. So that was his conundrum. So he just kind of put out the idea that there was some sort of toxin that they produced that could be spread around through the bloodstream that was causing problems. And that was kind of his idea. And it was very quickly discovered by a bunch of other scientists in Germany that this toxin did indeed exist. And you could grow up diphtheria bacteria in a lab, and you could purify this toxin away from them and use that toxin in the form of a syringe and then inject it into a guinea pig, and that guinea pig would die. Now, it would not develop a diphtheria infection. It did not come in contact with the organism itself here, this bacteria itself. It came in contact with the toxin. So the toxin was enough to kill the bacteria, or to kill the guinea pig, the toxin that organism produces. So a couple of people got to looking at this and went, I wonder if I can take this toxin here, and if I inject it in a guinea pig that's had diphtheria before, I wonder what's going to happen. So they had a guinea pig that had gotten sick from diphtheria naturally, and had recovered from diphtheria. Now, if you recall, that's one of our hallmarks of the secondary immune response, the adaptive immune response. It has to be able to have already seen the bacteria, that organism, for it to work. And this was the key here. If you inject it in a brand new guinea pig that's never seen the toxin before, the bacteria, it's going to die. But if you inject it in a guinea pig that's already had diphtheria and gotten better, these guys have a secondary immune response. Both their innate and their adaptive immune system can come into play, and that's the key here. So these guinea pigs, once they injected them with the diphtheria toxin, that purified toxin, it didn't matter. They were totally unharmed. So the scientists, once again, had no earthly idea what was going on here, but they thought that there was something in that guinea pig's blood that was able to protect against the toxin inside of that diphtheria vaccine, that syringe that they were injecting inside of that guinea pig. And they termed this thing the antitoxin. Now, once again, they had no earthly idea what it is. They just knew it was something there. So to figure out what was going on, to see if they had this idea right, what they did was they took together some blood from a guinea pig that had recovered from a diphtheria infection. And then they took some pure diphtheria toxin and they mixed them together inside of a syringe and they shook it up. The toxin inside of the diphtheria syringe was neutralized by the antitoxin inside of that guinea pig's blood. And when they injected that new solution inside of a brand new guinea pig that had never seen diphtheria, it was able to remain totally healthy, which means that there's something inside of that guinea pig's blood that was able to neutralize the toxin that protected another guinea pig. And that was the key here to figure out what was going on. It was very quickly discovered that if they could purify away this antitoxin, whatever it was, from those cells, if they could purify that antitoxin, they could inject that antitoxin into a healthy guinea pig, and then they could inject, or give it a couple of days, couple of weeks, and then they could inject pure diphtheria toxin into that guinea pig, and that guinea pig would never get sick. So they were able to transfer immunity from one organism to another using the concept of those toxins, antitoxins things. So if you've ever seen the movie Balto, this is what this is about, a very famous diphtheria outbreak in Nome, Alaska, in the early 1900s, excuse me. A very famous movie made by Disney, and then I think they have a live action movie that's either coming out or has come out recently. So check that out, very interesting movie about how they dealt with the antitoxin, a little bit about diphtheria and things like that. But anyway, so they were very quickly, these toxins, these antitoxins and things, they kind of got a different name that you guys are probably more familiar with. Antibodies was given the term antitoxins, or term antibodies, and the toxin itself was termed an antigen. So we'll talk about what an antigen is here in just a second. Yeah, it's on there. So we'll talk about what that stuff is in just a second, but let's go ahead and get into what the specific immune system is. So once again, I mentioned that the innate immune system has the exact same response for every single pathogen that it comes in contact with. No different response, doesn't matter what it is, it's going to try to kill it the same way every single time. And the innate immune system has to defeat the pathogen the first time for the adaptive immune system to come into play. The adaptive immune system comes only into play the second time around. Second time and then from then on out, but the first time the adaptive immune system either has to win or you die. The first time around is called the primary response with the adaptive immune or innate immune response. The secondary immune response is caused by the specific immune system playing the second time around. So the cool part about the specific immune system as well is it can remember what it sees. It has a type of specific cell called a B memory cell, we'll talk about in a second, that has the ability to essentially just take a snapshot of diseases that it's seen, of pathogens that it's seen, and remember them forever. So it's kind of wasteful for the body to continuously train to continuously produce antibodies to fight against malaria, if you haven't seen malaria in 40 years, or smallpox or whatever. chickenpox. So eventually what happens is the body will stop producing antibodies for those things in such a high number. But if you ever come in contact with it again, it will store what the information of how to make antibodies inside of those B cells are, those B memory cells, and that's the concept of amnesic memory. So if the body ever sees smallpox again, it could be 30 years later. You're not going to have any antibodies floating around in your body to help fight the infection, but your body will be able to make them rather quickly, as opposed to have never seen that organism before, and you have to make them from the very get-go. So B cells and T cells, this is how your body recognizes invaders and things like that. We'll go ahead and talk about antigens and antibodies and things like that. So antigens are things that are found on the surface of pathogens of pretty much every single organism on the planet that's going to trigger an immune response. So antigens can be pretty much anything out there. These are the antigens up here that is found on the surface of cells, mold, fungus, anything like that. That says, hey, I am a mold spore, I am a fungus spore, I am a bacteria. And essentially what happens is the body's going to use these unique antigens. I'll probably post a little video on this, a little more in-depth video on how this works. These unique presentations of antigens, each species has its unique antigen. They're going to use these unique little profiles to tell what species are. You have antigens as well, and your body reads those, and it's not a bad antigen, so it pretty much skips over those. If it sees an antigen or something that it doesn't like, it's the anti part. It's going to identify it and trigger the immune response to that. So antigens are very unique, like I mentioned, to each specific species. So each E. coli has, or all the E. colis have the same antigens on them all. Salmonella have the same antigens, kind of idea. So the way that particular antigen looks is called an epitope. And the antibodies inside of a cell are going to be looking for that epitope. So antibodies are specific as well. Antibodies, since the secondary immune response tailors its response each time, the antibodies for salmonella are only going to find the antigens for on salmonella. So they'll only find the epitopes that are found that fit salmonella, and that's the only thing that this antibody will work for. It will ignore all the rest of them, and you can kind of see that here. Anything that doesn't fit, it won't be able to bond with. So antibodies, we'll talk about the different types of them in a second. There are 5. They all pretty much look the same. They're known as immunoglobulins, or Igs, and you add another letter to it to give them their different types of antibodies. So this is the structure of an antibody up here. They're basically all the same. You can see down here we have our dark gray and our dark green. And these are something called the consistent regions. This is kind of like a key, if you like. This is the round part of the key and the long skinny part that they don't cut. And these are the different parts, the different cut part of the key, the different grooves and lines and things of the key, grooves and ridges. The consistent region never changes. This is what makes an antibody an antibody, much like what makes a key a key. That's why it's called the consistent region. The variable region up here in the top changes for each particular pathogen. That's the key part that changes for each individual door. That's why it won't unlock all the doors, even though it is a key. This is an antibody, but it is not the antibody for salmonella. The variable regions up here will have, say, a triangle for salmonella. They'll be shaped like a triangle that fits the triangle on salmonella's surface, or a circle like that fits the circle on E. coli, or a square or whatever that fits the square on Klebsiella or something like that. So that's how these things work. The consistent region makes this thing an antibody. The variable regions are going to be different for each particular type of pathogens. So you have the 2 heavy chains here that make up the consistent regions. The heavy chains on the inside, the 2 green ones are the heavy chains, and the 2 little gray ones are called the light chains. This is variable region up here is also called the antigen binding site, where the particular things are going to be different, and where the bacteria themselves or the little pathogen are going to be held. Now, essentially how this thing works is once, okay, so let's see if I can go back a little bit. So let's pretend it's our pathogen, or it is. So our pathogen's floating around inside of the body. Once this antibody is going to bind to this pathogen, you can see it finds the antigen that it likes. Once this antibody binds to this pathogen, a couple of things are going to happen. When an antibody binds to this antigen, this pathogen can become inactivated. Enough antibodies can stick to it that it will gum it up, it will overwhelm it, it won't be able to move around, and essentially it's just waiting to die. Once an antibody binds as well, the immune system then can be aware that that cell is there. The antibody binds, and this is essentially like waving a big giant red flag for the immune system to say, hey, come kill this cell, it's a problem. I've already identified we have a problem, which is why we have an antibody for it in the first place, and I found another one of these problems, come help. So once an antibody is bound to the cell, this cell's fate is sealed. It will be killed at some point in time. The antibody does not kill the cell. It just triggers the cell to be killed at some point in time by the immune system. It does not do any killing itself. But imagine if this guy was floating around in your body the very first time, and your immune system had to find him. This guy went and found him already and was waiting for him at the street corner before he even showed up and alerted the entire immune system that he was there. And then he went out and found all of them and handcuffed them to the street corner before they even caused a problem. So having an antibody response is a very good thing. The more antibodies in your body, the more likely you are to not have an infection or to have a less severe infection. So the different types of antibodies, they all pretty much look the same, but the most common one is something called IgG. So antibodies are going to be floating around in your bloodstream, and if they come across any pathogens, that's essentially what they're going to do. They're going to bond to it, which triggers the immune response. These guys are able to cross the placenta. And they, like I said, they make up about 80% of any immunoglobulins, all of the antibodies found in your body at any given time. So since they can cross the placenta, they give the baby, so the newborn fetus, the developing fetus, a slight bit of immunity for about 2 months or so from diseases that are found in the environment. Now, a long time ago, not so much a hard, fast rule today, but a long time ago, a couple hundred years ago, wherever you were born, it was a fairly high likelihood that your parents were also born there, and a fair likelihood that their parents were also born there. So whatever diseases that your great-grandparents encountered, there's a fairly high likelihood that you were going to encounter it as well. We all know that babies and infants and things are more likely, more susceptible to diseases. So if your mother could give you a slight edge of immunity, give you a little bit of immunity for the developing baby, for the diseases that are common in the area, it'll kind of tide you over for a couple of weeks. Until you can start making your own, a couple of months until you can start making your own immune system. Your body started to have a functioning immune system for itself, and you can start making your own antibodies. So this is just kind of a tide you over until your body can take over for itself. So a little bit of memory or a little bit of antibodies is kind of given to the baby to jumpstart them. Now, unfortunately, the body has never seen the pathogen, and that's the key characteristic for making more antibodies. Somebody gave you $1,000, you don't know how to make money, you don't have a job. So once the money's out, once the antibodies are used up, you don't get any more. You never came in contact with that pathogen, which is the key of how to make this secondary immune system work. You just got a free $1,000, you don't have a job. You never actually came in contact with the pathogen. So once again, it just binds directly to viruses or toxins, and sometimes it can kill them directly or neutralize the toxins, and it gums up the bacteria by sticking to them and keeping them from being able to move around, which then triggers the immune system to come over and just easily eat them up. So IgM is going to be turned to pentamer. So how these things work is the antigen binding sites down here. Each antigen binding site, this particular thing is called a dimer. There's 5 individual antibodies stuck on it. You can see they're all the little same shape Y. These little antigen binding sites here, there's one here, there's 5 of them, or 10 of them, 2 on each antibody. This antigen binding site can hold one bacteria, one organism, one. So this particular antibody can hold 10 organisms in place at the same time, can stick together 10 bacteria. This one can do 2. So IgM is going to be the very first type of antibody that's produced once your body realizes it has an infection. And IgA or IgG is going to bind, your macrophages are going to say, hey, we have an infection. And then IgM is going to be produced fairly quickly, but it's really expensive to make. You can see that here. But essentially, this is bring out the big guns. We don't want to spend a lot of money making little tiny guns when we can spend the same amount of money making big guns. So that's what it does. But you don't produce a ton of them because they are quite expensive. You come out hard, hit the infection really fast with a bunch of antibodies, and that's the idea. Once IgM is bound to an organism, it initiates that complement cascade and try to poke a hole in it. And that's the idea. This is also the very best antibody that triggers phagocytosis. Once this antibody has bound to a bacteria, that's the concept. Once it's bound, the antibody doesn't do the killing. It just tells the immune system to kill it. So once the antibody is bound to a bacteria that sticks to it, it tells the immune system to come over in some white blood cell or whatever to come eat it. So it's a really early detection system. IgA. IgA is going to be found in breast milk, saliva, and mucous membranes. This is going to be found alongside that beta-lysine, the lysozyme, and things that are found in the other innate immune system in those areas. It's a dimer. It can hold 4 bacteria, so 2 little Y's put together. So 2 bacteria in each little Y here, or one in each little Y, excuse me. So anyway, they last about 12 days or so, and they're abundant in eyeballs, nasal mucus, and things like that. And they are spread through breast milk, like I mentioned, so they can keep breastfed infants slightly protected as well from gastrointestinal pathogens and things like that. Same concept as that IgG through the placenta, but through breast milk instead. Now, a couple of bacteria and organisms have developed enzymes and things have evolved ways to degrade IgA so they can get around it in the immune system. So if they come in contact or they land on you on your eyes and things like that, they can easily get around IgA and still cause infection. IgD, we don't really understand how IgD works. It's found on the surface of B cells, but we don't really know what it does. It's kind of how these guys work with memory, but we don't really understand. So IgE, this is the guy that triggers your allergy responses, all of your allergic responses. So basophils, once these guys bond, this is what basophils are going to come attack. Your basophils and your eosinophils are going to be initiated and activated by the fact that these guys bond to something. So if these guys bond to something in a large enough numbers, the mast cells, the basophils, the eosinophils will come rushing to that area and release all their histamines and serotonins, and then welcome to an allergic response. So these are, once again, the different types of antibodies. You can see them down here. They all look the same. So the best way to tell them apart is where they're located at and what they do. So I'm going to post a video on how antibodies are formed. It's a little complex to go through in word form, so I'm going to post a visual video so you guys can see how this is worked through. It's a fairly easy process. The antigen is going to be detected for the first time by the macrophage in the body. This is our innate immune system. The macrophage is then going to take that antigen, that unique marker for the pathogen, and display it on the surface of its cell. Now we have B cells that make antibodies. Now B cells, they have lots of different antibodies on them. Some of them are going to fit, some of them won't. And essentially what's going to happen is a B cell that fits is going to bind to this antigen as well. So once the antigen over here on the B cell binds, the antibody on the B cell binds to the correct antigen, that antigen will then be removed and added to this B cell as well. So what you're looking for in the immune system is to have a macrophage with an antigen on it and a B cell that contains an antibody and the same antigen on it as well. So both of these guys in the immune system have found the same pathogen. And that shows to the body that it's not just a fluke. It is something to be concerned with. More than one cell have found this. It's something to be concerned with. So once a macrophage finds the antigen and the B cell find the same antigen, this is where helper T cell comes into play. This is how HIV prevents the immune system from working. It's going to kill this cell, these helper T cells. So once a helper T cell comes into play, it's going to lock with this B cell. It's going to lock with this macrophage. And it's going to determine, yes, this is the same antigen. Yes, this is the same antigen. And once it determines that it's the same antigen, it will activate this B cell. And once this B cell becomes activated, that antibody will then, the B cell will then become something termed a plasma cell, and it will start producing the same antibody that's found on its surface. So that antibody up here, that's the correct antibody for this antigen, will then be replicated and duplicated by the thousands and pumped out into the body, into the bloodstream. That antibody will then travel around through the bloodstream and stick to bacteria, which then triggers macrophages to eat them and other parts of the immune system to eat them. One of the other types of B cells, like I mentioned, is a memory B cell that will just hold that antibody on him and carry him around. If you notice over here, he lacks this little yellow thing, the MHC complex, so he cannot change the antibodies that he carries. He's stuck with that one, so he can start making more and converting into a plasma cell at any point in time. And we already have an antigen or an antibody made for this particular antigen. We don't have to go through this process anymore ever again. So I'll post a video, a little more complex video on how this works. Once again, HIV works by killing this helper cell. So if you can prevent this B cell from being activated, no antibodies will ever be produced. And that's how HIV works to kill the immune system. So once again, here's a little bit of a more graph of how that works. So I'll post a video again on this one. So lymphocytes, and we'll go ahead and lock this up just a little bit with our common protozoal infections. So Giardia, very interesting little protozoan. These are eukaryotes. It kind of looks like a little opera mask. These are suction cups that it uses to stick through the wall of your intestines, just like this. This is the intestinal wall. Tons of little Giardia stuck to the intestinal wall. And essentially what they do is they just sit there and they suck out nutrients through your intestinal wall. They suck out a little bit of food that you eat through your intestinal wall. Very, very, very painful. And it's spread by getting them through infected water. It's very often found in animals. A bear defecates in the water upstream. The little giardial cyst or the adult giardia go downstream. You take a drink of the water. Oh, it's a nice clean stream. Take a big old gulp. A couple of days later, the cyst or the active adults have woken up inside of your intestines and started to replicate and set up shop. You have some really violent diarrhea, horrible gas, horrible stomach cramps, horrible nausea, just awful, awful pain as these things sit there and do their job. And it lasts about a month or so, a little over if you're unlucky. And then your body can naturally shed this infection. It's kind of neat, and it'll eventually just go away. And they can use metronidazole, a very common antiparasitic drug, to help spread this, to help shed this one as well. So once again, they're going to spread by fecal oral. You're going to defecate in the water, or some sort of animal is, and you're going to drink that water in. And they're going to also diagnose this by looking for the living organism or their cysts that are spread through your feces in a stool sample. So trichomoniasis, very common STD, probably the most STD in the world that nobody knows about. This is a little organism up here. It has two flagella, which is one of the diagnostics of these guys. It swims around. So you take a pap smear from a person and you put it underneath a microscope and you look for little guys that are swimming around. It either means you are pregnant, congratulations, or you have one of these guys living inside of your urethra, inside of your vaginal canal. Not so good. Pretty much doesn't do anything at all. It can last for quite a while before it starts to show symptoms, up to years at a time. Sometimes it takes a couple of months. It's apparently just itchy, and that's about the only symptoms that this thing causes. If it gets serious, it can cause strawberry placenta or strawberry cervix. This is what this looks like down here. This is the hole that the sperm swims through to enter the fallopian tubes, and these little red spots are the parasite living on the surface of the cervix. It can cause some serious problems. Vaginitis, infections of the vagina, uretitis, same thing, infection in the ureters. It can cause slow birth weight and preterm delivery, but that's about really all of the problems that it causes. Nothing super serious. Very easily treated, once again, with that very common antiparasitic metronidazole. Super, super, super common as well. And our last little bit is malaria, Plasmodium vivax. A very interesting little intracellular parasite. It lives inside of red blood cells. These are the red blood cells up here, and you can see the little parasites curled up inside of the red blood cell. It's spread by the bite of a mosquito. So the mosquito takes a blood meal from someone who's infected. It sucks up the little mosquito parasites inside of those blood cells. It will then fly to a new human, take a blood meal from that human, pass some of those infected blood from the mosquito into the human, and that's how the infection takes over and is spread from host to host. Fever, vomiting, headache, coma, seizures can lead to death as well if this thing is not treated. Anti-malarial medication, very unfun treatment. You can prevent it by taking quinine pills. It causes the little parasite to not be able to invade the host cells quite as well. spread by the bite, once again, of a mosquito, and super, super, super common around the world at one point in time. Still very common in sub-Saharan Africa, parts of Indonesia, Asia, and places like that, in the tropics. Very, very, very common and causes quite a lot of deaths around the world every year. Essentially, what happens is you lose the ability to breathe. These guys kill your red blood cells, and you start having all the associated things with lack of oxygen, and eventually you'll fall into a coma, severe seizures, and eventually you will die. So that's all I got for this PowerPoint, guys. If you have any questions at all, please feel free to email me. If not, have a good rest of the day.